scientist Louis Pasteur once said, "Le hasard ne favorise que l'esprit prépare." Chance favours the prepared mind. Imagine life without the microwave, without Teflon, Post-its, and chocolate chip cookies. Serendipity has led to many interesting discoveries, and my introduction to science was somewhat serendipitous. As a young girl, I considered many career options, from vet and architect to Spice Girl and Pokemon trainer. But scientist, which brought to mind images of crazy old coots with fuzzy grey hair and glasses, never seemed like an attractive option to me. I stumbled into science, much like Alice stumbled down the rabbit hole and my eyes opened to a wonderland. I discovered that science is not about lab coat wearing loners 100 years ago who shut themselves away only to emerge decades later shouting, "Eureka!" Science is what propels the human race forward. It's an exciting world full of quirky characters where childlike curiosity and creativity are valued. All that's missing is the talking cat. In the wonderful world of science, anyone can find their niche. I'm fascinated by drug discovery, not the kind of questionable substances that may or may not be present in this guy's pipe. I'm talking about pharmaceuticals. These days, the focus is on rational design based on an in-depth understanding of how a disease works. But many important discoveries have been serendipitous. And often, the word isn't Eureka as such, but that's funny. Many of you will know the story of Alexander Fleming, who noticed a cell-free halo surrounding an area of mold that had contaminated his bacterial culture plate. His prepared mind and curiosity allowed him to isolate the substance that was killing the bacteria, which came to be known as penicillin. I find there's a certain satisfaction in knowing how a drug molecule works. In the presence of penicillin, a bacterial cell grows and grows until eventually, bam, the cell wall ruptures and the contents are spilt. But it took many years of dedicated scientific research to discover that the penicillin molecule was acting as an inhibitor, a plug of sorts, blocking an important enzyme that is essential for bacteria to survive. Think for a moment. What comes to mind when I say the word enzyme? Some of you are possibly imagining hungry little Pac-Men running around your washing machine eating up stains, but this image does not do them justice. Life as we know it would not exist without enzymes. Humans are made up of trillions of cells, and each cell contains thousands of different enzymes. They are molecules, usually proteins, that are exquisitely designed to catalyze the thousands of chemical reactions required for an organism to survive. The long chain of amino acids that makes up the protein molecule folds into a particular shape, bringing different parts of the molecule together to form a small pocket called the active site, into which a smaller substrate can bind. The shape of the active site is critical to its function, and substrate molecules of a particular shape are able to bind to the active site and react. The penicillin molecule works because its shape allows it to bind to the cell wall transpeptidase enzyme irreversibly, preventing it from doing its job. This enzyme is responsible for linking the molecules that make up the cell wall of bacteria. Without these links, the cell wall becomes structurally unsound and the bacteria cannot survive. Understanding how this antibiotic works has opened up a world of possibilities in terms of drug design. By studying the minute detail of an enzyme-catalyzed reaction and learning about the structure of a target enzyme, a molecule can be designed using chemical synthetic techniques to act as an inhibitor, binding tightly to that enzyme whilst leaving all others alone. Of course, the process is not as simple as just designing a molecule that inhibits a particular enzyme. The interaction of the molecule with the patient's body must be carefully tested, and the molecule must be modified in such a way that allows it to get into the cell where it is needed, and inhibit the enzyme without being toxic to the patient. Now, I'm sure you all have heard of antibiotic resistance and so-called superbugs. It all sounds very dramatic, but what does it really mean? Over time, bacteria may develop resistance to the effects of an antibiotic. In the case of penicillin, bacteria have evolved an enzyme called a beta-lactamase that is able to destroy the structure of the penicillin molecule by cleaving the beta-lactam ring, destroying its antibiotic properties. This has led to an arms race between bacteria and medicine. The bacteria evolved new mechanisms to provide resistance to antibiotics, and the scientists must come along and develop new drugs to overcome these resistance mechanisms, and so on. So we need to limit the use of antibiotics, as the more exposure that bacteria get to an antibiotic, the greater the likelihood they will develop resistance. Antibiotics, when used in the correct dosage, will overwhelm the bacteria, but when they are overused or when a patient cuts their dose short, the resistant strains have a chance to multiply and spread. Every time we prescribe antibiotics unnecessarily provides an opportunity for bacteria to evolve resistance. This is something that is crucial for the public to understand. Antibiotics are wonderful, but it is possible to have too much of a good thing. That being said, enzyme inhibitors can be used to combat a wide range of diseases, including tuberculosis. Now, tuberculosis is a devastating disease that kills over 1 million people worldwide each year. It is caused by a bacterium, Mycobacterium tuberculosis, and treatments are available, but multi-drug resistant strains have been discovered, and there is an urgent need to develop new pharmaceuticals to target these drug-resistant bacteria. Enzyme catalyzed reactions occur in sequences called metabolic pathways. Such pathways are used to do many things like breaking down carbohydrates, synthesizing hormones, or forming the cell wall of bacteria. The enzymes of the shikimate pathway, which leads to the synthesis of some amino acids, are attractive targets for antibiotic drug design. The shikimate pathway is essential for the survival of bacteria, such as Mycobacterium tuberculosis, but it is not present in humans because we don't need it, because we get those compounds from our diets. This is vital as the aim is to kill the bacteria, not the patient. This is exciting research that has been conducted in New Zealand. Investigations into the enzymes of the shikimate pathway, the exquisite detail of their structure, the mechanisms that they catalyze and their interactions with other molecules has led to the design of potent inhibitors that have potential use as antibiotics against tuberculosis. This guy here is 3-deoxy-D-arabinoheptylosonate 7-phosphate synthase. It catalyzes the first step of the shikimate pathway. And researchers at Canterbury University were able to develop inhibitors for this enzyme in the nanomolar range. This means that only a teeny tiny amount is required to prevent the enzyme from functioning. This is good news if you're a patient not wanting to take huge doses of medication, or a pharmaceutical company wanting to minimize costs and maximize profits. So, why should New Zealand invest in science? The public needs to know where the nation's money is going. If we fund 3 kidney transplants, we know we'll get 3 kidney transplants. But it is much harder to justify funding science. Remember back to my discussion of penicillin. This was discovered by accident, and Fleming was simply curious to see what it was that was killing the bacteria. And this led to the discovery of antibiotics, one of the most important discoveries in medicine, something that was totally unexpected, but something that has had an immeasurable effect on human health and the economy. The research into why penicillin works and extensive research into enzymes and the nitty-gritty detail of how they work has led to pharmaceuticals for a wide range of diseases. Good researchers are passionate about their respective fields. Some people, like me, are just curious to see how stuff works. Science is fun. It's a way of satisfying that inner child that's always asking, but why? But it is important to take a step back and look at the bigger picture. There is this perception that scientific research is some sort of luxury. This attitude must change. Science is absolutely essential to the growth and development of the New Zealand economy. If New Zealand can play a part in solving global issues such as tuberculosis, then the benefits to New Zealand society will be astronomical. Some of the largest contributors to the New Zealand economy are research and technology-based companies, such as Fisher & Paykel Healthcare. So an investment in science is an investment in New Zealand's future. Now, there are several valid approaches to scientific research, and they all have their benefits. Someone needs to ask fundamental questions, research driven by curiosity. Someone else may come along and take these discoveries and turn them into products. Yes, it requires money to be spent, and sometimes the financial benefits may not be immediately obvious. But it is crucial to remember that sometimes the most important research comes to those that weren't looking. We also need to stop being so possessive of our graduates. Successful scientific research requires collaboration, as science is so huge that one cannot possibly hope to do it alone. There's talk of a so-called brain drain. What if, instead of focusing on keeping our graduates in New Zealand, we encourage our researchers to spend time overseas, experiencing different research environments and forming invaluable international connections, opening up a possibility for greater collaboration? What if we work on making New Zealand a more attractive environment, a place that foreign graduates dream of visiting, a place that our graduates can't wait to return to, not just for our lifestyle, our scenery, or because Lord of the Rings was filmed here, but an attractive academic environment with top quality science, which is expensive, but the payoffs are huge. Make New Zealand a place of cutting-edge research, a scientific wonderland where the most creative, talented, and enthusiastic researchers can come together and make new discoveries, new inventions, and explore new ideas. New Zealand scientists have what it takes to compete on an international stage. All that's needed are the resources. Thank you.